We are here at Lincoln West School of Science and Health. All right, we are here with the plant smoke group. They are using pothos plants to look at the effects of cigarette smoke on the growth and health of these plants. All right, do you want to tell us your name?
Solimar Flecha.
You are using pothos plants here. What are you trying to learn more about?
Plants' reactions to tobacco, specifically tobacco water, tobacco just like smoke and stuff. Like how bad it is bad for like people in general.
What's been the most difficult part about the project?
That would be smoking the plants. We had trouble figuring out at first, especially because of that right there. I don't really know the name of it, but it couldn't work at first. So we had to use like actual fire with paper and stuff to smoke it at first.
That's great. So you figured out a different way to do it. That's awesome. What's been the most fun part of the project for you?
That would be like watching the plants' reactions to the tobacco, especially the smoking part. That's like mainly the most fun part. Like watching all the like smoke go into the plant and seeing like how it's like over time just wilting and slowly dying, and seeing how that would affect us the same way, and we just don't see it that way.
And last question, has this made you more interested in science careers?
It has, yeah. This whole thing is just interesting and fun. I like how we like did a hands-on and we got more involved through the project too.
We have a group that is interested in looking at the effects of alcohol on worms.
So last week we transferred the worms to the plates and add bacteria. Bacteria feeds them. So then we labeled the plates basically. And what we did was use two liquids. One of them is vanilla and the other one is alcohol to each corners basically. And now we're just checking to which way the worms travel, if they like vanilla more or alcohol, basically.
A group that is interested in isolation and depression. What are you using the worms to learn more about?
Isolation and depression. Period.
That seems like a big question to be asking. What's been the most difficult part of the project for you?
Touching them for real. Because I'm scared of worms. I ain't gonna lie to you. I ain't gonna lie to you.
What have you learned from doing this work?
Patience for real.
Yeah, well, I could use a little bit of that too, so I appreciate it. Alex, what are you working on for your project?
It's like a coffee experiment.
And what do you want to learn more about coffee?
How hyper can fruit flies get, and how hyper people can get.
What have you found in the results so far when you're feeding the caffeine to the flies?
They mostly die from the higher dose of the caffeine we put in than the lower dose over there.
The lesson is moderation, right? All right, Sean, can you tell us a little bit about your project?
Yeah, so basically what my project consists of is basically like experimenting on fruit flies and putting them on the screens to see like which one affects them more, I guess, something like that. But more so probably like seeing like how much damage it causes to like the brain off which screen, like the scrolling one, how long do you leave it on there? Like, how long are they effective for it? How many days could they survive? Something simple like that. I think she left them on there for like, I want to say like 5 days or maybe like a couple of weeks or so. And they did not survive. Like, they like literally probably just died. They looked like zombies. Like, they just literally just died. Like, I don't know, it's light, but like, I thought they'd probably try to get away from it instead of like going towards it maybe, but I don't know. It's just crazy. I think all the screens were bad just in general. I think they just all died. I thought the one that just stayed regular probably wouldn't kill them, but I guess all of them did. They just couldn't stare at the screen for that long.
Have you ever worked with Drosophila before, the fruit flies?
No, this will be my first time.
What's been the most fun part of the project for you?
Probably like separating them, so like now I could tell like their gender, like off just looking at them, like the separating part to understand like which one's a male, which one's a female, something like that.
When you've been doing this project, has it made you more interested in science?
Yeah, it made me more interested just experimenting because I like hands-on stuff more than just like writing on papers and stuff. So this was fine. I liked it.
Chad.
Taylor. Mashaila.
All right, everybody. Can you tell me a little bit about your project?
So our project is how music affects our mind and what would be more compatible for like children in like schools areas or like when you're trying to put a baby to sleep. So I wanted to do this because music is my favorite subject and I like music, and it's going to be like helpful because like I know like most babies, they like different types of genres, and I was kind of really curious about that, like why does babies like different kind of genres, and so that's what I did. I kind of put it into what I liked and did it with flies.
They seem to go to the top away from the music, which means I'm assuming that they don't like the music or they're not fond of it. So I believe that has some type of effect on humans as well, if they're having a negative effect on it.
Yeah, we have noticed that heavy metal and the classic music has been killing the bugs instead of the meditation.
Oh, wow. So we think maybe that they enjoy the meditation, or at least doesn't kill them.
Yes.
Last week there was like gook inside the headphones and it was so bad. I was like laughing and just like trying to clean it off because it was fun, and it was like interesting to see that happened over 3 weeks. Like all the debris from the tube and the flies went onto the headphones. It was really fun and cool.
Can you tell us a little bit about your project?
So what we're doing is we're setting up each groups and putting CBD, which is drugs, into the tubes and seeing how they affect each fly and how may die or survive, or if there's any after effects. Well, we've had most of them die all the time. Most of them, even without the CBD, they die most of the time. But even with the CBD, most of them do survive, but most of them don't. We think it's because of overdosing on the food.
Has this project, has it made you more interested in science?
Mm-hmm. Yeah, I want to learn more about like animals and how different things affect them from doing this.
Okay, so we chose to do sleep paralysis to see how does the sleep affect male flies and female flies, also by age.
And how did you come up with the idea for the project?
I came up with sleep paralysis because I experienced it, so I wanted to see how would it affect somebody else, as in fruit flies, if they was to experience sleep paralysis.
What's been the most fun part of the project for you all?
Doing little races, preparing the flies. We're preparing to dissect them to see any differences between the females and males inside their brains.
Okay, we're testing caffeine, the effect on caffeine on worms, and like we dissected them, we fed them, and then like to test the effect, we time to time they barreled versus the low caffeine, the controlled, and the high caffeine. Lower caffeine they were more bigger than like the higher and the no caffeine, which is kind of odd. What's your favorite part?
Feeding the worms.
And killing them.
It's an experiment about crabs and marijuana, and it's how marijuana affects the human body. And with the crabs, it's just showing us like the different actions that they have and how it messes with the brain. Like right now we have a dissected crab and we're trying to test the brain activity and see like how it affected the brain.
And we use young crabs to show how marijuana is affecting the young youths of these days. So yeah, and we see how they grow with the CBD.
Like they getting lost places. Like they like moving around too much, getting lost.
Yeah, we have had a lot of death scares because the marijuana crabs do not move at all.
So
and it's like,
yeah. But once they get in the box with the camera, they start moving around.
Yeah. Control grows like faster than the CBD, and we noticed that one also.
R.I.P. crab number C.
Yeah.
Our biggest
R.I.P.
crab letter C.
I saw this box over here that you have set up. What is going on in there?
There's crabs in
So
there.
this is the line-in box, and the test is to see. So we put all the crabs, we line them up, and we tend to see like what scares them and what doesn't, and see if like the marijuana crabs, and see how active they are, and see if the marijuana affects the crabs.
So we're doing like
or
a
doesn't.
hawk. So we have a picture of
We changed it into flashing lights because
we have flashing lights, and it's like to see how it affects them. So right now, those are the CBD crabs, and we're trying to see if the flashing light scares them, and then we're gonna do it with the control and see like the activity, the difference in the activity, and like to see like if the marijuana makes them calmer, and it's like. For the control, we're going to see if they're just naturally calm or if it actually scares them.
Yeah, because last time we did it over here, they came out of their shell and actually started crawling and stuff, and we was all surprised. Yeah. And we all took pictures and stuff. Everybody was just surprised and happy. Because they don't come out their shell, but when they by themselves, yeah.
So our project is we're putting UV light, we're exposing UV light to flies. And today we are extracting protein to see if the UV light really worked and affected the flies.
It's a really interesting idea. How did you all come up with the idea for the project?
So I wanted to be a pediatrician, and I wanted to see how different diseases affect children. I'm very confident about my future because this project helped me see what causes different diseases in children.
So the main focus is to figure out what fertilizers work well with the plants to see how they grow and which ones work better, basically. Just which ones work best with the plants and see how well they grow to see which one we could use for growing plants.
What has been the most fun part of the project for you?
I think trying to germanize the plants, seeing how they'll sprout and then planting them to see how they'll grow and seeing the roots and stuff.
We're doing the study, the social behavior of autism. So we're using C. elegans to study. As of right now, we're studying their social behavior or any changes they've had, and they've had some changes so far. Socially, some are not socializing and some are socializing a bit too much.
The C. elegans change, for example, on the 14th last month, we did things if they're how much they clowned by 10 out of 10, and some were one was 100, the highest was 180%.
What led me to do it is, okay, so autism has a spectrum, and autism is more, you can't really tell if someone has it or not because it's one of those things that can be hidden or it can be pushed out there. So I want to start this project to see if we can really find it and see if it's easier to diagnose and help people that actually have it.
What's been the most fun part of the project for you all?
I think it's been looking at the C. elegans and looking into a peep of their life to see what's happening.
We're doing social anxiety with worms. We're watching their behavior, with, putting them in different kinds of concepts, like the sun and the dark and refrigerators, and yeah, we'll see how they react.
We are doing a project on sleep deprivation on flies, and at the end we're going to cut their heads open and look at their brains.
What's been the most fun part of the project for you?
Decapitating them.
Yeah, cutting their heads off.
Yeah.
All right, Maria, can you tell us a bit about your cockroach project here?
Our cockroach project is, everybody think that roaches just eat anything. So we did 6 different foods to see over time about if they eat it or not, or how much they ate, and if they grew or if they lost weight because they wasn't eating. And we have a control. She is big, really big. We fed them raw burgers. They was frozen though. And we got some vegetables. We got carrots and cauliflower, some corn and green beans, and pineapples and mango.
What has been the most fun part of this project for you all?
Watching them grow.
Feeding them. They be hissing and they be running everywhere.
Yeah, we did just hear one hissing, so just dealing with the model organism has been tricky for you all?
Yes, sir.
Yeah.
Some of them grew a lot from the first time that we measured them and we first did the project.
The first time that we met them, half of them was little, and now that we've been feeding them, they kind of got bigger. So.
Which diet made them biggest?
The male corn and green beans.
Oh, okay.
10.3 grams.
We're working on blackworms and planaria and how they regenerate in certain conditions. One of the conditions we have is we're using this chemical called FGF. It is to help things grow faster in the natural world. And while this is going on, we are seeing if it actually does work and if it does grow faster, or in our last test, it seems that the planaria have seemed to not grow or have been stunted in their growth since they have been introduced to FGF. Another thing about the blackworms is that they actually seem to grow a lot longer, or they're a lot more colorful than their normal non-FGF variants. Right now we are testing if the resinoric acid actually works and if they do specific things to this planaria or blackworms. And that is what we're doing right now. How could we observe something that has to do with limb growth? So they pick planaria and blackworms as they have the natural ability to regrow their body parts.
Right.
And we were trying to figure out what would happen if we observed and we could figure out why they grow and how these acids or chemicals help them grow or allow them to heal faster than they normally do in the wild. The most fun part of the project is probably the ability to talk with others to figure out what is going on, how everything is going, and what is really going on with these.
Our project is about like plants and putting alcohol in it. Does it affect their growth speed or how fast they grow? And like, they're like cells and stuff. The ones with less alcohol grow faster than the ones with alcohol. I feel like that's because the alcohol is like poison to them, so it's not really helping them grow. Getting the like exact measurements and stuff is pretty fun to me. Measuring was like hard at first, but as you like moved on, you got used to it, so it's not that hard anymore.
Okay, we are here with Miss Beard. She is one of our teaching partners this year. We wanted to hear a little bit from you about the impact that the program has had on some of your students.
Oh, it has had a positive impact. Students that typically are not engaged are so very engaged. They're writing. They want to come in and check and make their observations when they are not in class. It has been wonderful, and they are always asking when you guys are coming. Actually doing hands-on, students that typically maybe aren't engaged in lab and class because they're shy or they're unsure of themselves are getting more confident in measuring and participating, making observations, and other students are helping them along as well.
Great. Well, we appreciate you being a teaching partner with us.
Thank you. We appreciate you guys.